In this section, I'm going to talk about the history of microbiology, and then I'm going to talk about the scientists that contributed to that history. So early ideas about disease transmission, people knew that certain foods spoiled and became inedible or caused illness. If you think about foods that have spoiled, like let's think about milk. If you open up milk in your fridge and it has spoiled, you can smell it and it smells disgusting. We have developed that ability to smell foods that are contaminated with microorganisms and not want to eat those foods. So we have that ability now because of evolution. Black plague and smallpox is caused by some kind of transmissible matter. So they knew that the black plague and smallpox could be transmitted from person to person. And then there was the belief in spontaneous generation, where microorganisms just appeared out of thin air. There were also beliefs of evil spirits. A lot of people thought that if you got a certain illness, that you were evil. If you were sick, it was because you did something bad and you had to be punished for it. And then we established the scientific method. So early scientists tended to explain natural phenomena by a mixture of belief, superstition, and argument, but during the 1600s, true scientific thinking developed. And the scientific method starts with hypothesis, then prediction, experimentation, results, and conclusions. Hypothesis is just an explanation of something in the natural world. Prediction is talking about predicting why you think that occurs. Experiment, you would test your prediction, and then, or test your hypothesis, so you would test what you were thinking happens, and then report your results, which you combine all that data and try to make meaning of the data, and then conclusions. You would come up with a concluding statement. When did we start studying microbiology? So the ancient Egyptians used to keep moldy bread in the house to rub on wounds. We already talked about that. We know why they did it was because it prevented the growth of bacteria. Grapes used to be used to make wine. In 1020 AD, tuberculosis was suggested to be infectious. In 1546 AD, epidemiology was explored, and all of these were suggestions until Robert Hooke observed the cell for the first time. So Robert Hooke in 1660s observed the cork, and he coined the word cell. So when he looked at cork, cork is plant material. And if you've ever looked at plant cells under the microscope, they're all rectangular, and some of them are square-shaped. Because they have a rigid cell wall, they look like squares or rectangles. So when he looked at that, he actually thought it looked like a prison cell. So he called them cells, and that's why we call a cell today. Antoine van Leeuwenhoek in 1684 developed a single-lens microscope. He drew the original drawings of cell shape and is known as the father of bacteriology and protozoology. When you are tested on these scientists, know their major contributions, but you do not have to know specific dates or times that this occurred. Carlos Linnaeus in the 1750s developed binomial nomenclature. Binomial. Bi means two. Nomial is talking about a name. Two-name naming system. Nomenclature, naming system. So we have Escherichia coli. We typically call it E. coli, but that E stands for a word. The Escherichia is talking about the genus name, and coli is the species name. Just like Homo sapiens is the genus and species name for a human. Louis Pasteur in 1860s. Louis Pasteur discovered chirality, pasteurization, that microbes were in the air, and investigated vaccination. So Pasteur liked to heat up liquids and see what happens. So he wanted to prove that microbes were in the air. So what he did was he developed this swan-necked flask. So he heated up this swan-necked flask in an effort to kill everything inside of it. And then he kept it upright. And for years, the solution in here stayed normal. It stayed uncolonized. Because anything that were to settle from the air was trapped down in this location, and it wasn't able to make its way up into this broth solution. But as soon as he tipped it over, in a matter of hours or days, microbes grew and spoiled the broth. So that demonstrated that there were microbes in the air, and that if you prevent air from entering into the broth, then the broth would stay uninfected or would stay essentially sterile. With pasteurization, so pasteurization is how we in the United States preserve our milk. So we heat the milk to a certain temperature for a very short period of time, and that kills most of the microorganisms in the milk. Way back when, tuberculosis and brucellosis were major infectious pathogens that were killing tons and tons of people that were drinking milk directly from the animal. But through the use of pasteurization, we decreased contamination and the spread of tuberculosis and brucellosis through the milk. And that was all just based on the research that he did in order to develop this system to successfully decontaminate the milk. In 1870s, Robert Koch developed Koch's postulates. So basically, they used to still kind of think that maybe infectious diseases could be transmitted from one person to another, but a lot of people still thought evil spirits were abound and that there was spontaneous generation where people just suddenly got sick with something that came out of nowhere. Koch's postulates demonstrated for the first time that something from an organism, so whether it's a mouse or a person, can be transmitted to a healthy organism and make that organism sick. So he started with this mouse, and the mouse was dead. So he looked at the blood of the mouse under a microscope and noticed these organisms. And then he put that blood of the mouse and plated it on an agar plate and allowed it to grow. Then he scraped some of the growth off of the plate and injected it into a perfectly healthy mouse. And that mouse too died. And when he looked at the mouse's blood and looked at the plate of the stuff that grew from that mouse, it was identical to what grew and what was found in the blood of that original mouse. So he demonstrated for the first time that yes, there is a disease progression and microorganisms can be spread from person to person, and that there is an organism that's causing the disease, not an evil spirit. In the 1920s, Alexander Fleming discovered penicillin, and we'll talk more about the discovery of penicillin and Alexander Fleming when we get to antibiotics. Robert Whittaker in the 1950s came up with a 5-kingdom classification system. So these may be familiar to you: animals, plants, fungi, protists, and Monera. So Animalia, Plantae, Fungi, Protista, and Monera. This 5-kingdom system no longer is commonly accepted in the scientific community. So now we've replaced the 5-kingdom system with the 3-domain system. So the 3-domain system was developed by Carl Woese in the 1970s. Carl Woese used 16S rRNA typing in order to classify organisms, living organisms. So 16S rRNA, what is that? So rRNA is talking about ribosomal RNA. So that's what makes up the actual ribosome. And with the ribosome, there's a small subunit and a big subunit. So what Carl Woese did was got the sequence of the ribosomal RNA of the small subunit. And that sequence was so unique that he was actually able to investigate the differences and the similarities in that genetic makeup there, the makeup of the ribosome, to classify organisms. And initially when he did this, everyone laughed at him. He was disbarred from scientific communities. He was a laughing joke. And after his death, everyone said, hey, wait a minute, that was actually a pretty good idea. And now it's commonly accepted as one of the best and most accurate ways to classify life. So we have 3 domains: archaea, bacteria, and eukarya. Archaea, as we talked about earlier, are like bacteria in that they're single-celled, but they don't have peptidoglycan in their cell walls. Bacteria do have peptidoglycan in their cell walls, and bacteria and archaea lack the nucleus, while the domain eukarya has a nucleus. So if we look at evolution of life, we can see that bacteria are over here, and archaea are actually closer to eukaryotes than to bacteria. Eukaryotes include plants, animals, fungi, and protists, and then Monera, that 5th kingdom, would be the domain bacteria and domain archaea. Archaea are all microbes, bacteria are all microbes, and eukarya, some microbes and some regular organisms. So in 1970s, restriction enzymes were discovered. The 1980s, the PCR technique was investigated. 2000s, the importance of small RNAs was elaborated on. And 2010s and beyond, genetic identification of the human microbiome is in the works. So we want to know every microorganism that lives in the human so that we can associate it with certain disease conditions. Science is an ever evolving collection of new information. All of these things we will talk about eventually in this course. So we're going to talk about restriction enzymes, PCRs, or PCR techniques, genetic identification, and the importance of the human biome. We're going to talk about all this stuff as the course progresses, but new discoveries are made continuously. There's actually a new technology where they're trying to get the whole genome sequenced of a microorganism in order to identify the microorganism and determine which antibiotic would be effective to use against that microorganism. So things are rapidly evolving and we're constantly developing new technologies.